we are going to go over the 4 basic vertebrate tissues. So we're going to go over all 4, look at characteristics of them, talk about location, and also some ideas on how to prepare for a test regarding these tissues. So these are the 4. The first is epithelial tissue, and this is just kind of a diagram of just a generic epithelial tissue. And you can see that the cells are stacked very close together. So here is a cell right here where my pointer is, and there's the nucleus, and right next to it on this side there's another cell, and there's the nucleus, and on the other side of it there's a cell, and there's the nucleus. And so these cells are very close together. At the bottom, they're all being anchored down on this protein basement membrane. So they're all anchored down on this. And then underneath that, they have a connective tissue layer. So this right here is a connective tissue layer. Above it is going to be the free or apical surface. Now, these are found covering different parts of the body, either on the outside or on the inside of the body. So you take your skin, for example, your skin is covered by an epithelial tissue, and that tissue basically protects your skin and your body. On the inside, you have coverings of epithelial tissues, for example, in your tubes that make up your body, like your blood vessels. Within the blood vessel lining, you have an epithelial covering. In your digestive system, you have epithelial tissue lining. Inside your sweat gland tubes, you have a lining of epithelial tissues. So they cover things. That's a basic idea of them. And I said that underneath that basement membrane was a layer of connective tissue. So we're going to look at connective tissues in a very general sense next. These connective tissues are being shown by this diagram here. And what you see is just a combination of different tissues here. You have blood, you have fat cells, you have this cell right here, which is making fibers. That's called a fibroblast. You have white blood cells like this one, which will help with your immunity. You have these fibers that are these pink ones right here and the blue ones right there. And basically it's an array of different cells and fibers, and these cells are far away from each other. So that's the thing to realize with the connective tissue is it connects, it binds things together. The cells are not as closely packed as with the epithelial tissue because it's not serving as a barrier or as a membrane. Rather, it's serving as a way to connect tissues above and below it and to the side of it together. And it's a very diverse family of tissues, and so we're gonna have to see what they look like in detail one by one to make more sense of it. The third type of tissue is muscular tissue, which is being depicted by this picture right here. And what you see are skeletal muscle cells in this case. And these skeletal muscle cells have, they're long and skinny, and they can contract. So the thing that's important about muscular tissue, if you were wondering why muscular tissue is important or is unique, it's because of its ability to contract. And what does contract mean? It can scrunch up and get shorter. And if it can scrunch up and get shorter, that means that if it's holding or attached to 2 things, one on one side and one on the other side of its length, and it scrunches itself up, then those 2 things can move. Now, we typically want to think about bones moving because of our muscles contracting. So if I've got a bone on this side and a bone on that side, and the muscle scrunches or changes its length, those two bones are going to move because they are attached to the muscle via tendons. Now, we do have other types of muscle tissue other than skeletal muscle tissue. We also have muscle tissue in the heart, known as cardiac muscle tissue, and also in your visceral organs, and that's called smooth muscle. We're going to see those in more detail. And the fourth type of tissue is known as nervous tissue. And nervous tissue is the tissue that is required for all the other tissues to do what they need to do, because you need a wiring that basically tells things to react or to move or to do things. So just like the wiring in your house gives the electricity to have a light bulb shine light, or to turn on your toaster, or what have you, or to give power to your television, the nervous system made up of neurons like this one is going to be the one that sends electrical signals to effector organs or tissues to get them to do something. So in other words, if I want this muscle over here to contract, I need to send a signal to tell it to contract. It's not going to do it by itself. And this cell right here can send that signal to that muscle and get it to contract. So now what we're going to do is we're going to go over each category one by one and look at examples. And we're going to start with epithelial tissue and look at first the cells that make them up. So if we want to make a continuous sheet that lines different parts of the body, we need to basically put the cells close together in rows like this, or like that, or like this. But the cells can have different shapes. And so what we found is that they can either be cube-shaped, and we call those cuboidal cells. They can be flat pancake-shaped cells, and we call those squamous cells. Or they can be long and skinny, like columns, and we call those columnar. Now you'll see in front of each one, each shape, it has the word simple. So simple cuboidal, simple squamous, simple columnar. And that just means, well, in this picture, I've got one row of cuboidal cells. So that's why I call it simple cuboidal. Here, I've got one row of squamous cells. So I call it simple squamous. One row of columnar cells, so simple columnar. Now, if I have two or more rows of these cells, I will call it stratified. So in this case, it's a stratified squamous layer. And it may look cuboidal down here, and so you might be asking yourself why it's called squamous. Well, we label it based on the very topmost layer. So the very topmost layer is flat and squamous. So there are many rows, and some of them may be different shapes, but the topmost layer gives us our shape name, which is the squamous. And stratified just means that we have more than one row of these. In all these cases, you do have the basement membrane that the bottom row is attached to. Okay, so here is one of these looked at in detail. These are columnar cells. And these columnar cells, because they're tall and skinny, they look like columns. That's why we call them that. Here you have the basement membrane, and underneath you have the connective tissue. On top, you have what is called the apical surface. Think of the word apex as the word top, okay? And the apical surface is important because, well, if you're thinking about your skin, the apical surface is making contact with whatever is outside your body. If you're thinking about the apical surface in your intestines, well, that's going to be making contact with whatever food you're trying to digest. If you're thinking about the apical surface of your epithelial tissue in your blood vessels, well, that apical surface will be making contact with blood. So the apical surface is the free surface, the surface that has something on top of it, right? And so that's why that's important, and that's going to vary significantly as to what the function of the apical surface is, depending on the location in the body. So here is a type of epithelial tissue. You see the one row of cells right there. And you see the basement membrane that's highlighted here, right here in green. Now, this is called pseudostratified. And the reason why it's called pseudostratified is because it sometimes looks stratified, but it's actually one layer of cells. And we're going to see in detail how that works when we look at the specific example. But this is just still in general, just talking about epithelial tissues in general. So just a couple more things regarding just general terms. Think about this one right here. This is a simple layer, one simple row. This is the pseudostratified, which it looks like it's stratified. So if I look at this little triangle there, it looks like that might be part of a different row compared to that one. However, notice that it's touching the basement membrane, and notice that this taller one is touching the basement membrane. So they're all touching the basement membrane, and so they're all part of the same row, actually. That's why we call it pseudo-stratified. It's not really stratified. And then this one right here is definitely stratified because you have the first row touching the basement membrane, the second row not touching it, the third row definitely not touching it, and so on and so forth. Here's your pancake shaped, or some would like to think it looks like a fried egg. Either way, whatever helps you remember this stuff. We call that squamous. There's your cuboidal, cube-shaped. And here's your columnar or column-shaped cell. So let's look at a real example of this, and we'll go over these one by one. So the first one that you have to know is one of the easier ones to make out. And we're looking inside the kidney, which is the organ that makes urine for us. So there's the kidney. And this kidney, it takes the blood that's coming in through this red artery right there. It takes that blood, and you can see it here. If you take a slice of the kidney, you can see that there's that red artery system that goes in. But then adjacent to it, what you see are these yellow tubes. And those yellow tubes are called kidney tubules. Kidney tubules because they're tubes after all, or sometimes we call them renal tubules. Now, if we take a slice of one, or two, or three, we're going to see, if we look inside the hole, or the lumen, we sometimes call the hole of a tube a lumen, of that tubule, what we'll see is the lumen. That's the lumen right there. But making up the tube is a single row of cells that is in a circular formation. So notice that this is one row of cells, right? Because watch how I trace the basement membrane. There it is. There's a continuous piece of basement membrane, right? And so here is the first cell in the row. Here is the second cell in the row. Here's the third cell in the row. And here's the fourth cell, and fifth, and sixth, and seventh, which tells you something about when we say simple, it doesn't mean it has to be a flat row. We can fold it into different shapes. But as long as all the cells are touching the basement membrane, it's one row. Because you could imagine slicing it right here, okay, and opening it up that way, and turning it into one row of cuboidal cells. So this is simple cuboidal epithelium. This is the apical surface, right? And in here would be the fluid that eventually turns into urine that is being made by these, what we call nephrons of the kidney. So here are some images that you need to get ready for regarding the test. So you need to be able to see what kind of depictions are drawings or computer animated productions, and then what kind of pictures are actual real histological slides. So this is a drawing pretty clearly. I don't know if you can see that. Maybe you need more practice with it to realize that, but that's not real, and neither is this over here, right? This is computer animated. This one may be computer, I don't really know. It could be computer animated, maybe it's drawn, but it's not real. These, on the other hand, are real. So these are histological slides. And what we mean by that is we've taken real life tissue. We've sliced it into very thin slices. We've stained it with a dye that helps certain structures show better. And we've put it on a glass slide. And then we look at it under the microscope. And so this right here is the basement membrane. And it's making this circumference around it. And then here is a simple cuboidal cell. There's another one, there's another one, there's another one, there's another one. And this is the lumen. So this is the tubule, right? This is the tube. And we've just taken a slice of it. So you got to imagine that this thing keeps going, right? It's not just right here. It's going to be a tube. So you can imagine it going into the screen and out of the screen. And then this is the one right next to it. So that's a really good histological picture. And if we're lucky, then we'll get one that looks like this. And that's good. If we get one that looks like this, then no problem. But we can't always expect them to be perfect. And so histological slides vary tremendously regarding how good they are, what quality they're in, and how easy it is to make out the structure. So on the right, there's another histological slide of simple cuboidal epithelium. And notice that it's the same structure. So there it is right there. We're a little bit zoomed out, so that means we have more tubules here. There's one right there. There's another one right there. There's another one right there. There's another one right there. And so you can see that there are several of them in this picture just because we're at lower magnification. And the image is not as good, but I hope that you can realize that the image on the left and the image on the right are depicting the same thing. And my advice is always to Google search simple cuboidal epithelium or whatever epithelium you're trying to memorize, and hit images and try to see a whole array of different types so that you start learning the structure in a variety of ways instead of just learning it in one picture and memorizing just that one. The next epithelium that we're going to look at is simple columnar. And what we're looking at is the small intestine. And if we take the small intestine, we take a little bitty slice right there in that little box. And we look at that slice, what we'll see is a what we call a villus, which is a fold that arises right there. And if we magnify it, there it is. And on each villus, we're going to have a row of columnar cells going around it like that. So it's hard to see in this because it's a smaller image, but that's a cell, and that's a cell, and that's a cell, and that's a cell. And it's making its way around this villus. And each one is a columnar cell. This picture is a bit more clear. So here's a villus. The plural is villi, right? So villus is the singular, villi is the plural. So it's pointing to two of them, so that's why I call it villi. And notice that you're going to have a connective tissue core in here, which is not being depicted here, but we're going to see on the slides. But the lining, because epithelia are all about lining, there's a cell, there's a cell, there's a cell, there's a cell. The lining is made up of these simple columnar cells, and each one has these microvilli on it, which are smaller versions of villi. And microvilli are for absorption for each one of these cells. The villus is for overall absorption in a big way. And the microvilli are for these micro absorptions that each cell has to do. And so very much like a sponge, the small intestine is going to absorb nutrients using these villi and microvilli. So here is an actual picture of it. So realize that the previous slide was not a real picture. and this is the actual one. And so here is the folding of that intestine. There's the fold. You can see it folding there. You can imagine kind of folding again up that way. And there is a simple columnar epithelium, a columnar cell, another columnar cell, another columnar cell, another columnar cell. That means that this right here is the basement membrane. Now, what you'll notice is these clear cells right there. Those are known as goblet cells. Goblet, as in what you drink of. You drink out of a goblet. And these goblet cells are going to secrete mucus onto the lining, right, onto the apical surface. So the intestines do secrete mucus in order to let the food that you're trying to digest move along the tube. It acts as a lubricant. Okay, so here's another picture of a villus. There's the fold right there. And all of this in the core is connective tissue. And there is a columnar cell, and there's another one. And right there, there's the goblet cell. You can always tell the goblet cells because they look clear. And you can really see that, you can see the mucus right there. And over here, you see these little balls of mucus that are being manufactured by these cells. And these cells secrete the mucus onto the lining. Now, what you cannot see very well are the microvilli, but what you do see is kind of a shaded apical surface, and that shaded apical surface is shaded that way because of the microvilli. So that gives you a sense of where they are. They're too difficult to see with this magnification, but trust me, they are there. Okay, so the next thing we're going to look at is pseudo-stratified columnar epithelium. And this is famously found inside your airways. And specifically, we're going to be looking at the trachea. The trachea is the windpipe that takes the air that you breathe in to the lungs and takes the air that you breathe out of the lungs and out of your mouth or out of your nose. Now, notice that this trachea is in front of the esophagus. There's the esophagus right there. The trachea is in front, and if you've ever felt your throat, you know that there are some kind of some hard parts to it, like some bumpy pieces on it. And those bumps that are hard are the cartilaginous rings that protect the trachea and keep it open, so that air can constantly flow in and out of this tube. Because if it collapsed, you can see the esophagus is kind of collapsed. In fact, it does tend to collapse unless you are eating and you swallow, and then the food kind of opens it up again. But typically, the esophagus is collapsed if it's not being used. The trachea, on the other hand, is forced open with these cartilaginous rings. And so here's a slice of it, and there's the tube. And this is the cartilaginous ring, this horse-shaped, this U-shaped structure. And it looks like a horseshoe shape, right? And this is the lumen inside the trachea. And then the question is, well, what is the epithelium made of right there on the inside? And so here is a view of that horseshoe-shaped U, just a little slice of it, so it's kind of curving like that. This is the cartilage, and on the inside this is the lumen. So on the inside, you're going to have this darker piece that's making the covering or the lining, and that's the epithelium. That is pseudo-stratified epithelium. And we're going to see it in more magnification to see the detail. This is from far away, so you can't really see it very well. But in the next slide, we're going to see magnified pseudo-stratified epithelium, and you're going to see that it also has cilia, which are really cool. So here it is. This image on the left is showing the cartilage, and this right here is the pseudo-stratified epithelium. And you can really see that that little brush on top are the cilia. Now, these cilia help to move the mucus around because we're going to have goblet cells in here too. If you can see that right there, that's a goblet cell, and that's a goblet cell. So you can see they're kind of clear looking. They're going to be making mucus as well, and you know that when you get sick, you're making mucus. This is one of the defenses that you have against microbes. So when you're trying to fight off an infection by making more mucus and putting that onto the epithelium, well, that's just an extra coat of covering that microbes have to get through in order to infect your tissues. So it's a very, very useful way to fight off infection. However, you don't want the mucus just to sit there, and so you need to get rid of it. And so these cilia are going to beat and move and basically clean out that mucus. So you secrete the mucus, and then the cilia move it out of the way, right? And this picture kind of shows you, okay, you've got this pseudostratified epithelium. There's a small cell. There's a bigger cell. And on the very tip, right there, you've got the cilia on that pseudostratified membrane. So just remember, the trachea has pseudostratified epithelium with goblet cells. The next group of cells we're going to look at are connective tissue cells. Connective tissues are the next group of tissues we're going to look at, and there's a big variety of them, and they may look rather different, but they all share certain characteristics. So we're going to look at the characteristics first. The cells are sparse, and this just means that they're further away from each other typically, right? So I'll give you an example here. You have that's a cell, and that's a cell, and that's a cell, and that's a cell, and these cells are far away from each other. Now, these stringy structures are proteins that the cells make, and there is a extracellular matrix or fluid that all of this is bathing in. And so that's a big characteristic of connective tissues. Now, in some cases, you might see the arrangement is different of these fibers. So that's a cell right here, and that's a cell, and that's a cell, and that's a cell. You can tell by the nuclei that those are cells. And these cells are called fibroblasts, and they are making these protein fibers, which are these pink bands that are going in these waves in parallel right here to each other. So just realize that in this case, the fibers may be closer together, but the cells are still pretty far away from each other. In this case, you have more space in between with whatever extracellular matrix is going to be there. But the idea is that there's a lot of fibers in the tissue, and there is this extracellular matrix, and the cells are not as close together typically compared to the epithelial tissues that we have seen. So this image on the top is called dense regular connective tissue, and it makes up your ligaments and your tendons, and you can see that the collagen fibers are going in the same direction, and this makes the tissue very strong. If you tug on it in this direction, it's going to be able to resist that tension. So that's why it's a good tissue to make your ligaments and your tendons. This one right here is a softer type, and you can tell it's softer because the protein fibers are not as densely packed with each other, which means that it's going to be more delicate. And this is known as areolar connective tissue, which is a type of connective tissue that sits underneath a lot of epithelial tissues as a kind of binding, structural, connective support structure. So not nearly as tough as what's going on in your tendons or your ligaments, but nevertheless does give strength to all sorts of even visceral organs, etc. Another example is adipose connective tissue, which is right here. And so these are adipocytes. And adipocytes are basically fat storage cells. Now you can see that there is this extracellular matrix in between them. Some of the cells do get close together, but you still have these gaps of space. And so it still fits into the connective tissue category. This is also going to be a supporting tissue that wraps around organs for protection, and it helps to bind things together, to hold things together, and it's also, of course, important for the energy storage of fat. So when you are running out of sugar, you need to go to your long-term energy store, which is your fat stores. And so these will either fill up with fat when you're nicely fed and you have more energy coming in that you need. You're going to fill these up with fat and they're going to get larger. Or in times of starvation, they will decrease their fat. They will release the fat, and that fat can go to cells that are working hard, and they can go into respiration for ATP synthesis for energy. All right, so just kind of an array of these different tissues still. So areolar connective tissue. You might see other names in different descriptions, but for us it's going to be areolar connective tissue. Dense regular connective tissue right there. Adipose tissue, fat tissue basically. Under the skin is a very important location for adipose tissue. Blood is a connective tissue, and again you can see the cells are further apart, and so there is going to be an extracellular matrix, and there are going to be fibers within the blood plasma. That fluid is going to have fibers within it that make up that connective tissue extracellular matrix. Now in some cases we get connective tissue extracellular matrices that are rather tough, like in cartilage. Cartilage is pretty tough material. It's the padding in your joints, so it has to have some sort of toughness to it. And even tougher is going to be bone tissue. And bone tissue is fully calcified, making that extracellular matrix really, really tough. But you can see here that the cells, which are these darker structures, are definitely farther away from each other than in epithelial tissues. And so what's in between them, all that yellow stuff, is a calcified extracellular matrix. But bone is also considered a connective tissue. So let's start with areolar. Here is a close-up of it. Notice that we have fibroblasts, which are cells that make the fibers for us. So they literally sit there and secrete these proteinaceous fibers that then build up this matrix and make it strong. Here are the collagen fibers, which are these big, thick bands. And then you also need some elasticity. And so you do have certain fibers that are for elasticity. Those are called elastic fibers. And what that means is that if this tissue stretches, then it can go back to its original shape without too much trouble. So we need to know where it's found. It's widely distributed under the epithelia of the body, forms the lamina propria of mucous membranes. That's the part that is underneath that simple columnar epithelium is going to be the lamina propria. And it's also going to be around organs and capillaries and so on and so forth. So it's kind of all over the place. And you need to know what it looks like, and you need to realize that, okay, I see these fibers, I see the cells, and everywhere in between is going to be the extracellular matrix. So again, what we want to do is look at a few different examples, because not every slide is the same. Remember, when we prepare a slide from a real organism and slice it and dye it, well, there's no way we can make the same slide twice. It's always going to be different. And that's one of the challenges of histology, but that's also one of the beautiful things about histology is that you do get to see that variety. Once you spend enough time with it, it really starts making sense. And so just realize that this slide right here is areolar connective tissue, and so is this one, right? And so try to see the similarities. Realize that there are some differences regarding the color tones and what pops out at you better and what not, but at the end of the day, the structure is the same. And this one right here is nicely labeled for us with, for example, white blood cells for immunity, mast cells which release histamine, again for immunity. You've got the fibroblast cell that's making the fibers. You've got the collagen fibers, the elastic fibers. So when it's nicely labeled for you, that's always nice, but just remember, on a test it will not be, so you have to be prepared for this stuff. All right, the next connective tissue we want to look at is dense regular connective tissue. And again, we looked at this already a little bit. You're going to find these in your tendons and your ligaments. So just remember that tendons connect muscle to bone. Ligaments connect bone to bone. But in either case, you need to have a very, very strong connection. And so again, we have fibroblasts, and we have fibroblasts releasing this collagen, and this collagen creates this rope-like structure that can connect this muscle to the bone or can connect another bone to another bone. Again, we want to look at some pictures to get a good sense of this, and here these look like they can vary quite a bit. Notice the big thing that you have to notice for this to realize it is dense regular connective tissue is the arrangement of the fibers, right? These fibers are all going in the same direction. So in this image, it's kind of faint. The stain is kind of faint. It looks like the nuclei, you can kind of maybe see some nuclei of fibroblasts, but not as easy to make out. But the collagen is there. It's going in the same direction. It's dense, and so it does have the properties of dense regular connective tissue. This one on the right is much more clear. You clearly see the fibroblasts. You clearly realize where the collagen is. So that's more clear. But don't expect all pictures to look like this one. Oftentimes we get one that looks like this. Also, the magnification is important. Right, so this one looks like it's further away at lower magnification. This one right here is at lower magnification, and this one right here looks like it's zoomed in better. And then also be ready for these slides to be flipped, right? So when we put the stain down on the glass, it can go in all different directions. And so just realize that it may not always be going in this horizontal way. In this case, it's the fibers are arranged in this vertical fashion, but still they're moving in the same direction, creating that rope structure that can connect muscle to bone or bone to bone. So the next connective tissue we want to look at is bone. And bone is the hard connective tissue because it is calcified. So calcium is what makes it hard. Also the collagen and the calcium together. The cells that you find inside the bone are called osteocytes. Osteo means bone, cyte means cell. And this is just a diagram of an osteocyte. And what you notice is that it is kind of in a little pocket, right? So there's the extracellular matrix that's calcified. So that's hard. That's hard, that's hard, that's hard, that's hard. And so there's like a little pocket within that hard matrix. And that's where the osteocyte resides. And look at it reaching out in between that hard matrix. And we call these little rivers canaliculi, which connect one osteocyte to another. So what are we looking at over here on the left, which is a real picture of it? Well, these darker spots are the osteocytes. So you have to imagine that this cell right here is that one right there. And then these stripes that you see, these lines that you see in the picture, are the canaliculi that connect different cells together, because these osteocytes want to share nutrients and materials and stuff like that in order to make this bone healthy. In the middle, you have a central canal, which is going to have nutrients coming in with blood. And those nutrients are then going to diffuse out through those canaliculi to the osteocytes that are all around this circular structure. So the circular structure is the big theme here when it comes to bone tissue, and that's known as an osteon. And so this is a close-up of an osteon, but if we look at this picture right here, we see that that right there is an osteon. This is an osteon. This is an osteon. So you can see repeating osteons. Each one should have a central canal. There's one and there's one, and there's one and there's one. Each one should have a central canal to go along with it. So here's a diagram of it. If we take a little slice, a little pizza slice of this femur, which is the thigh bone of the human, we see that if we take a slice of it and magnify it, we see these repeating osteons. So there's one, and there's the central canal with the artery and vein and also the nerve, and there's one as well. And so this is a kind of a classic picture of this, what we call the Haversian system. We call it the Haversian system because of these repeating Haversian canals. The word Haversian canal and central canal are synonymous. And then if we look at this little bit of the osteon and we magnify it, what we see are the osteocytes. And you can see how in this picture very clearly those canaliculi are reaching out from one osteocyte to the next so that these cells can share nutrients with each other. From the central canal, the nutrients are going to go out and diffuse out through these canaliculi to feed all of these osteocytes throughout the perimeter of this osteon. So let's look at some examples of pictures. Again, we want to always try to see some variety of pictures so that we get better at this. And so here number one is showing us, well, what is that? That right there is an osteon. Number two is here pointing to this center part. What is that? That's the central or Haversian canal. 4 is pointing to an osteocyte. And then 3, I didn't say this yet, but these layers that you see around the osteon, looks like layers of an onion. Those are called the lamellae of the osteon. And here's another picture of a Haversian system. Central canal, there's the first lamella, there's the second lamella, just like an onion. There's the fourth lamella. Some might say that it looks like tree rings, and I think it might, but don't get confused with your plant histology. Make sure you can tell the two apart. Now, in lab, what you're often going to see are models, plastic models, that depict this Haversian system. So this right here is the pizza slice, but it's in plastic. So you'll see these in the labs. And here you can see the lamellae. And what they've done here is, it's hard to see here in this two-dimensional picture, but they've kind of sliced it like a wedding cake, where you can see the lamella that's in the middle there poking out, and then the next lamella there, and they've just kind of chiseled it one layer at a time just to show it, but that's just for the model's sake. And so you can see how it's labeled here, but basically it's the same stuff that we went over in the diagrams. Different plastic models might have different ways of depicting this stuff. This right here is just a really close-up view of one osteon. And so this is the Haversian canal. Blue is the vein, red is the artery. Those are the lamellae. There's one, there's two, okay, there's three, there's four, right? So these rings that are going around this. And again, what they've done is they've kind of cut it at different levels in order just to show the structure in different ways in different places. The little pockets where the osteocytes reside in, so they're the osteocytes, right? They're these darker cells right there. But notice that they've kind of removed them in some locations, and they just left a little pocket where that osteocyte used to be, and those are called lacuna. And lacuna are the little pockets, or sometimes people call them pits, where these osteocytes reside in. And of course, what happens is when we find bone, real bone of an organism that has passed away and everything is decomposed, especially the softer tissues decompose. And so definitely you don't have osteocytes after some time of decomposition, but what you do have is the matrix that remains, and that matrix does hold on to these little pockets, and so you can, with a microscope, you can really see them well, and we call those lacunae. So bone is the hard tissue that composes your skeleton, your endoskeleton. That's your structural hard material, and it's calcified, and that's what makes it hard. But it doesn't serve for very good padding, and so in between bones, what we have are areas where we can move about, and those are called joints. And so, for example, you think about your knee joint, or your hip joint, or your elbow joint, or your shoulder joint. And these joints all need to be padded because the bones are getting really close to each other. And bone, if you've ever hurt yourself by hitting your bone on something, you know that bone is painful. It's innervated if it's damaged. And so what we do is we put this nice layer of cartilage inside the joint, covering the ends of the bones so that you don't have bone hitting bone. And that's one of the more famous reasons to have cartilage. What is less commonly known is that the fetal skeleton is composed of cartilage. And so as you're forming these bones, they're first made of cartilage, and then the cartilage gets replaced by this calcified matrix of bone as development continues. This allows for the fetus to be softer. You don't want a very calcified fetus because childbirth requires a lot of flexibility, and so you want to have a skeleton that's much more supple. Cartilage is much more bendy than bone is, so that's why that is useful. And basically, as we develop and get older, we keep losing cartilage, actually. So a 5-year-old will have a lot more cartilage than a 25-year-old. A 25-year-old will have more cartilage than a 65-year-old. And in fact, arthritis is a condition that we get as we get older, and that is because we lose cartilage over time, and what's left is bone, and when bone scrapes bone, it is definitely painful. Now, there are other locations for cartilage, so make sure you read up on those other locations and functions as well. So the cartilage we're going to look at is called hyaline cartilage, which means glassy cartilage. Looks like, I guess it looks like glass. And it's one of the prettier slides to look at. So it also has lacunae, which are these pockets or pits where the cells reside, but these cells are not called osteocytes. They are cartilage cells, so they're called chondrocytes. So the word chondro means cartilage. Cyte means cell. So cartilage cell inside the lacuna. And again, over here, you have another depiction of it. And what you notice again is that the cells are further away from each other. They're not as close as epithelial cells typically. And therefore, you're going to have this extracellular matrix that is in between them and binding all of this together. And that's why we call it connective tissue, because it's all kind of bound up together. So the next connective tissue we want to look at is blood. And we're going to look at the different cells that blood has. The cells that are in most abundance are the red blood cells, which makes blood red, and are the oxygen carrying cells. So when you look at this slide over here, most of what you see are red blood cells. Sometimes we say RBCs. A more scientific term is erythrocyte for a red blood cell. And so that's an erythrocyte, that's an erythrocyte, that one, and that one, and that one. And then down here you have different types of white blood cells, or a more scientific term is called leukocyte. Or sometimes we'll just say WBC for white blood cell. And these are the white blood cells here. So that's a white blood cell, and that's a white blood cell. Now the way to distinguish this, I think it's pretty clear, but just in case, the red blood cells do not have a nucleus, and so their center is rather clear looking, almost like hollow, almost like, not quite a donut, but almost like a donut. And the white blood cells, on the other hand, are larger and definitely have a nucleus, and sometimes you'll see these nuclei, they're kind of funny shaped with these multiple lobes there. And these white blood cells can vary, so just keep that in mind. And we don't have to really figure out which one is which for this course, but for more advanced courses, you may have to distinguish one from the other. What you cannot see too well, maybe there's a speck and there's a speck, but it won't be tagged on a test, are the platelets. It will not be tagged on a test just because they're too small, and so just to avoid any confusion, we typically don't tag them, but just realize that they're there, and these are important for blood clotting. That's how your blood clots, is with platelets. And the more scientific term, even though it's not here, they're known as thrombocytes. So again, we want to look at different examples. The zoom in versus the zoom out is going to make a difference, but you can see basically here, hopefully you can see that most of all of this are red blood cells, and the white blood cells are these larger ones that are going to have variety of them, right? And we don't have to necessarily point out which one is which, but just realize that those are the white blood cells. And so on a test, the pointer might be pointing to one of these, and you'd have to know it's a white blood cell. Here you can really tell, okay, that's a white blood cell versus that's a red blood cell, that's a white blood cell, and that's a red blood cell. You can really see how empty that looks because it does not have a nucleus. So now we're moving on to a new type of tissue, and that's known as muscle tissue. And there are three types of muscle tissue. There is cardiac muscle, there is skeletal muscle, there's smooth muscle. Cardiac muscle is found inside the heart, and it is involuntary, meaning you cannot control it voluntarily. You can't choose to contract these muscles. They do it automatically without your knowledge of it. Smooth muscle is found inside your viscera, So the way that you move food along in your intestines, for example, is by muscular contractions. The way you mechanically digest food in your stomach is through muscular contractions. And again, these are involuntary. And that means you do not have control of them. They digest on their own, right? You don't have to think about digestion. It just happens for you, which is nice. This one in the middle, skeletal muscle, is voluntary, meaning that you do have control of it. So you're typically choosing, right, to move an arm or a leg or what have you. And so because you have control of that, we call that voluntary. Now, cardiac, we call it cardiac just because the location is the heart. Skeletal, we call it skeletal because these muscles typically attach to bones of your skeleton and move them about. And smooth muscle, we call it smooth because notice that there are no stripes on that muscle. So skeletal muscle has these stripes, which we call striations. Cardiac muscle has striations, but smooth muscle does not. So because it looks smooth to your eye, it's called smooth. So cardiac muscle is characteristic because the cells are short and branched with one nucleus. So here's one cell with a branch going there, a branch there, and a branch there. Okay, and so these three branches here show that it's this one branched cell with one nucleus. And then these bands that are much darker than the typical striations are called intercalated discs, which is the mechanism for these cardiac muscle cells to communicate with each other and to contract rhythmically all together. So again, these are found inside the heart. These are involuntary, they are striated, and they have intercalated disc. A very common test question is to point the pointer onto the intercalated disc and ask you what that is. And so make sure you know that. Skeletal muscle has the striations, but the cells are enormously long. So one cool fact about skeletal muscle is that if you take a calf muscle, right, that each muscle, a cell, will be the length of that calf muscle, and they'll go in parallel with each other. And so this is just like a little window of this cell. This cell just keeps going and going and going. And because it's so long, it has multiple nuclei. So notice how many nuclei one cell has. So the characteristics for skeletal muscle are, it does have striations, it is voluntary, contains multiple nuclei. They do attach to bone. These are really long, skinny cells. Okay, and so one muscle, that tells you how long each cell is. And smooth muscle is characteristic because these cells are short and tapered. When I say tapered, I mean that each end of it is kind of pointy, spindle-like. And the center is a bit thicker. So you take any one of these and you look at it, and it should have that characteristic shape, right? So the center is kind of thicker with the nucleus in there, and then the end is tapered like a spindle on either side. And what you should not see are the striations. So this looks like a drawing or a computer animation of it, and this one right here is the real thing. And so please look at this one on the left for your test, because that's the one that it may show up, and you'd have to know that that right there is smooth muscle. Now, again, this is involuntary, right? It's found inside your viscera, so digestive purposes, this stomach and intestine have to contract in order to move things along or to mechanically digest food, but you're not consciously doing it, so it is involuntary. It does it for you. Okay, see if you can label these three. Do you know which one is which? These are the three different muscles. Maybe pause the video and try to figure it out yourself before I give you the answer. So this one on the left is the skeletal muscle. I can tell it's skeletal muscle because I see multiple nuclei. This looks like it's one long, giant cell. Here I see the striations. And so it's striped. And so that tells me it's skeletal. This one in the middle is cardiac. How do I know? Intercalated disc. So these darker bands are the intercalated discs. One nucleus per cell. So there's one nucleus there. But intercalated discs in between cells. So there's one cell, intercalated disc, intercalated disc. So that's cardiac. And this one is smooth, and how can I tell? I got to look at that tapering that goes on there, right? I don't see striations, and I do see this tapered appearance, spindle-like appearance on either side of that cell. Again, in lab, what we do is we also look at the models of these structures. And so try to figure out, based on your knowledge of the histology, which type of muscle this would be. Again, if you want to pause the video before I give you the answer, it might be a good idea. This is a smooth muscle model. How do I know? One nucleus, a spindle-shaped tapering on that side, a spindle-shaped tapering on the other side, a bigger center part. So, smooth muscle. The last type of tissue is the nervous tissue. This tissue is the one that sends the signals to all the others to tell them to do stuff. And it does so with electrical activity that we call action potentials. So this is a neuron, and neurons are cells that are found in your brain or in your spinal cord. And what they have are a thick portion called a cell body, and within the cell body is the nucleus. And from there you'll have these skinny branches called dendrites where the input goes in. So if I want to give this cell information, I got to go through the dendrites to give it information. So the dendrites are the input. And then this cell has to figure out what to do once it gets the input and send another signal to another cell. And it does so through the longer, thicker branch, and that's called the axon, right? So you can really see dendrites all around the soma there, and then a very clear axon over here. So here's another histological picture of it, and hopefully you can see, okay, that's a dendrite, 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 that's a dendrite. This is the soma. Soma is another word for cell body, by the way. You'll see it either way. There is the nucleus, and this branch right here is the axon, right? And this axon can be really long. So, for example, if this is a neuron in your spinal cord, and you need to send this axon out to your big toe, that's how long it's going to be. It's going to be the length from your spinal cord to your big toe, and that's how you get your big toe to wiggle about, is with a neuron sending the muscle of the big toe an electrical signal called an action potential. Rather cool. Again, in lab, what we're going to see are plastic models that try to depict this, and again, in 3 dimensions it's better, but we have to do what we must. In 2 dimensions we have to try our best. This is the cell body. What we've done is we've kind of removed the cell body from the rest of the axon there, right? So this is one cell. Just realize that. Okay, they've just sliced it right there and stuck this piece here. Okay, But just realize these two things are connected in actuality. So here's the cell body, there's the nucleus. These are the dendrites. These gray ones are the dendrites. Okay? The gray ones are the dendrites. So what are these orange things that are sticking onto them like suction cups? Okay, those are the axon terminals of another neuron, maybe somewhere over here, sending its signal to those dendrites. So that's where the signal goes from one neuron to the next. Is these are other neurons attaching themselves to the dendrites of this neuron, trying to give it some information. And so the job of this neuron is to take that info, right? It's going to take that info from the input coming in from this axon terminal and this axon terminal and this axon terminal, and it's going to try to decide what to do. And if it needs to send an action potential, it will, and that action potential will go through this axon here, which remember is connected here, right? They've just sliced it, and so the information would go down through this like a wire, like an electrical wire moving this way. The material that insulates this wire is made by a type of cell called a Schwann cell. Schwann cell. So notice that this right here is the nucleus of a Schwann cell. And this Schwann cell secretes this layered myelin sheath, which is a type of coating that wraps around the axon. So notice that this center part is the axon, but this right here is the myelin sheath. And what that does is it insulates the electrical signal. It makes the signal also travel much faster down the axon. Now, here is kind of a physiological picture of how all this works. You take a slice of the spinal cord, just a slice of it, right? You're just kind of looking at the spinal cord as if you slice it, and you look inside of it. The cell body of the neuron starts somewhere there. It's going to exit this way with its axon. It's going to travel through a nerve and go all the way to a skeletal muscle cell that is part of this larger big muscle here that is attached via this tendon to that bone, and it's going to innervate or give it the signal to contract. And when it gives it the signal to contract, these skeletal muscle cells can scrunch up and get tinier and move the bones about. And so the model that is typical in lab is this one, which goes with this picture on the left. So notice that this right here, right, that's a Schwann cell, sliced right there. That's another Schwann cell, right? They look like sausage links, kind of, right? And this has been sliced right here. So this would be much longer, right, moving all the way along, all the way to the spinal cord in that direction. And here is the connection that it's making to the muscle. And so notice that this right here is that synaptic end bulb of the axon that is attaching to the muscle, just like nerves attaching to nerves. Nerves also attach to muscle and tell them to contract. And that's what's happening here. Right here at this point, that is the neuromuscular junction, or the point of attachment of the nervous system with the skeletal muscular system. And when this neuron tells this muscle to contract, it's going to scrunch. And when it scrunches, it's going to move the bones that it's attached to about. And so this right here, therefore, is the skeletal muscle cell itself, or sometimes we call it muscle fiber.